you.
Okay. I was like, I don't think I saw you. We should talk about how to get things taken care of.
Okay, sounds good.
Does anyone have any questions to start us off?
Can you go over the noggin and then the BMP4?
Yes. Let me grab a slide because that's probably the easiest way to talk about it. So, the three tissue types, so we have ectoderm, endoderm, mesoderm. The differences between those tissue types are that they have receptors, okay? So if you have a certain type of receptor, now you can be that tissue type. So we've gone from totipotent to pluripotent. And the how we are pluripotent is we have a receptor. In ectoderm, we have the receptor TGF-B, TGF-beta. That can receive a signal. And when the signal is received, it makes skin. So the default state of ectoderm is neural tissue. But we're going to make a lot of skin. So we're going to make it whenever we have BMP4 present. If noggin is present, it binds to the active site, preventing BMP4 from binding. Therefore, it turns into neural tissue. So noggin makes your noggin.
Okay.
Okay, so this slide's a really good slide illustrating that. Now, how do you make mesoderm? Mesoderm is a different receptor, right? Have we gone over that receptor?
I think so.
We have not. We did not go over that receptor because different receptors will behave very similarly, right? So we're using this example. We're not using all of them. We haven't gone over everything. But endoderm has different receptors. Mesoderm does. That's what made them pluripotent.
So the TGF receptor, that's on the ectoderm?
Correct.
Okay.
And that's what's telling it to do what. And so ectoderm has that receptor. Endoderm has a different receptor. Noggin is released by what? Let me know what noggin is released by.
Would it be initiator protein?
No.
Is it BMP4?
No, because BMP4 is what noggin's blocking. Noggin is released by Henson's node. In the mesoderm, Henson's node releases noggin. That's why we create this circle shape. So in the worksheet, okay, 1 second, let me share a different screen. I'm on a different computer and this is not, okay. Give me a second. Oh man, I hate. There we go. It's in a different spot. So we have our 3 tissue layers. Okay. And so ectoderm's on the top, mesoderm's in the middle, endoderm's on the bottom. And we have a set of cells in the center here, right here where this little dot is. That's going to release noggin. So noggin is going to move through diffusion in these directions. And when it gets to that spot, that's where it's going to make neural tissue when it comes in contact with that receptor. So we have this round shape because of diffusion. Why isn't noggin working in mesoderm?
It doesn't have the same receptors?
There we go. Why doesn't it work in endoderm? Doesn't have those receptors, so it only works where the receptors are there. Now, BMP4 is everywhere, okay? And that's what's going to make skin be the thing that's expressed mostly. All right. Next question.
Could we go through the process of how it is expressed, like in what steps?
What do you mean by that? What
Sorry.
is expressed?
The noggin and BMP4.
Okay, so express just means that it's been turned on, right?
Mm-hmm.
So we are transcribing a gene. That's it. Noggin is a protein. To make noggin, you have to have that transcription, that regulation. So why do only a few cells in Henson's node express it?
As the signal?
No, it's the ones that have been regulated. It's been turned on. Everybody else is turned off. So regulation is what controls transcription. Okay? So if I'm going to make noggin, I need to turn it on. So we're going to regulate it. If I want to turn noggin off, I shut it off. And you can shut it off by turning off the operator, or you can turn off, you can histonate it, you can methylate it, you can do any number of different things to shut something off. But to activate it, you need to be able to transcribe it. Okay, so we're going to regulate it, so it's going to be transcribed. That transcripts are going to make in translation, a protein. That protein is called noggin. It's going to bind. It's being made in Henson's node. It's being made by a few set number of cells in the center of the body.
And then that moves to the ectoderm?
Diffusion, it moves to the ectoderm. It moves other places too, but it's only working in places that have the receptors. There you go.
From the worksheet of noggin, it says on number 5, why is neural fate limited to the midline of the ectoderm? Why doesn't all of the ectoderm become neural in a normal embryo?
Okay, so what did we just show about diffusion? Where is noggin being expressed? Endoderm, ectoderm, and mesoderm?
Mesoderm?
Is being expressed in the mesoderm. Excellent. Where at in the mesoderm?
Where the receptors are?
No, there's no receptors for noggin in the mesoderm.
At Henson's node?
At Henson's node, excellent, which is in the center of
The mesoderm?
mesoderm. So we've set up our center axis. Okay. 1 second. I have a diagram that might actually be of help. So if it's in the center of the mesoderm and it's being expressed there, it's transversing everywhere. And you guys have had physics yet or talked in chemistry about Reynolds number?
I have a question. Are you showing something or not?
Not yet.
Oh, okay.
So you talk about Reynolds number. Reynolds number is diffusion, okay? It's talking about the diffusion constant. And so things diffuse out from that center line. And so when you're an ectoderm, from the center is where you're going to get the expression pattern. So that's where the signals are going to come from. We'll talk about this this is an example I'll show you guys on Monday. I put it in the slides today. If we look here, we have the nodes, so the three-dimensional view. So it's moving down the streak, but it's also in the center, right? So it's the center line. And so as it's moving down, that expression is happening. right? And then this would be a three-dimensional view of the notochord. Does that help? So you have the center mesoderm is where it's being expressed, and it's going out and it's radiating out.
Are
And when
you
it comes
are
in
you
contact of ectoderm?
are you pointing, because I can't see where you're pointing? I'm sorry.
No, just I'm walking through the steps.
Okay.
And as it moves out through the ectoderm, if it has the receptor there, it will bind. If it doesn't have the receptor, it won't bind.
And so the entirety of the ectoderm doesn't have it, and so that's why it doesn't go.
The entirety of the ectoderm has the receptors, but it does not receive the signal noggin.
Okay.
Okay, so let's go to a blank slide so I can annotate. Okay, so man, this shit does not work on this computer. All right, so here's your receptors. Okay, they're everywhere, right? But the signal is being released right here in the center. As that signal radiates out, it will only work in the area where it has the receptors and the signal. So where I'm shading in. Okay, so the signal is being received everywhere it's radiating out, but it'll only work where the signal can be received in the receptors. And so that's the cross-sectional view of the body, okay? We look at the top, like the embryo we looked at yesterday. Hensen's node starts here and works its way down. And so if we work in our way down, and remember this is a three-dimensional view, so not only is it working its way down, but it's radiating out. Hence, we're going to create this tube right down the middle.
Okay, and so it stops like towards the back, towards the posterior.
Correct.
Okay.
Oh, I know we didn't go over it, but like, is this kind of like the same thing as like in the book? It mentions like an MPF subunit, and in order for like the MPF to function, it needs the cyclin that's connected to the protein.
Sort of. That's not a receptor. That's a regulatory component, and we're going to talk about cyclin B and regulation on Monday when we start thinking about cell cycling in cancer.
Okay.
Did we go over immunology?
We have not yet. We're going to do that next week.
Okay.
We're a tiny bit behind.
That's
So,
okay. I just remember reading about the T cells, B cells, and
yeah, we're getting there.
okay.
We're going to do blood typing, T cells, B cells, immunology, cancer, cyclin C, cyclin D, all of that next week. Any other questions?
What are the somite defects?
Somite defects are, so what do somites make again?
I don't remember. I just know that they run through the body.
They do. So they run through the body and they're what creates segments. Somites make vertebrae.
Oh, okay. Yeah.
So when you have a defect in the vertebrae, that can give you anything from like scoliosis to Kleinfelders to any number of things where like the vertebrae fuse, you don't get rotation of axes, things like that. Another type of somite defect you could have is where the skin does not cover back up the neurulation, and in this case you would have exposed nerves. That's a little bit more rare.
I have a question about problem set 8.
What's the question?
So I have a question about number 3, and then how to interpret the schematic on number 5.
Okay, so we're looking at number 3 first.
Yeah.
Dolly, the sheep one.
Yeah. The permanency of differentiation. I mean, that, to me, I interpret that as like once a cell is differentiated, like it knows what it's going to be. It's always going to be that.
Correct, but it has all the genes to make everything else. So just like we showed in class, you can go from determined or differentiated back to totipotent.
Oh, because everything is spontaneous and reversible.
Correct. We can we can reverse things. We can, yeah.
That makes no sense how they took an adult mammary gland cell and put it in what?
So they've taken a skin cell. So they took an adult cell, okay? All the genes, all your genes are found in every single cell you have.
Mm-hmm.
Okay? But not all genes are turned on, not all genes are active, right? So if I take your skin cell, put it into an egg, an empty egg, I go from a determined back to totipotent, I can make a new one of you, correct?
Right.
That's what they did.
So is this saying they cloned it?
Correct.
Quote unquote? Oh, okay. Whose own haploid nucleus. So does that just mean their own chromosomes had been removed?
Correct.
Interesting. Okay. And then number 5, the schematic, I tried to interpret it. I interpret it as the dorsal as the ectoderm and the ventral as the endoderm. But it's just a bunch of circles to me.
So this is where it wants you to kind of put things. So you're having that 3-dimensional view, right? The cross section. And so if we look at the top of the cross section, so we go back to that worksheet that we just did, right? The noggin worksheet.
Mm-hmm.
Top is ectoderm, bottom is endoderm. Well, now I have a 3-dimensional structure. So the outside edge is the ectoderm. So what's inside, right? So we have our dorsal, then we have a tube within a tube. So you have not only your neurulation tube, but you have your digestion tube as well. That's what this is showing. So if I cut your abdomen in and create a cross section of it, that's what this is trying to illustrate.
Okay. Okay.
So yeah, just tube within tubes.
Gotcha.
Okay. Any other questions? Are we done? No other questions?
I have questions, but I'm trying to formulate them in my brain.
Okay. Anybody else with any questions? want to see my goob? Here she is.
Hello.
Hi, Sylvie.
She's
You hanging out?
We're just
Look at
hanging.
that big window. You seeing the trees outside?
And the view.
Ooh, you can see the mountains.
Yeah.
That's cool.
We live in the mountains.
Yeah, in the woods. Is that a river down there?
Lake.
Lake? Oh my gosh.
Yeah. We live on Lake Kavanaugh.
That's nice.
Can you explain, because I have cell signaling causes different transcription factors to be produced in various cells and embryos.
Correct.
Can you explain
So,
that?
okay, so what does totipotent mean?
Many?
Any. It can be anything, right?
Okay.
So if we're anything, do we have any signaling going on?
Yes.
No, actually.
No. Okay.
Because nothing has been turned on yet.
Okay.
Okay, so when we go from anything to many things, from toti to pluripotent, so we go from blastulation to gastrulation. Okay, so now we've formed our 3 tissue layers. Things get turned on. Okay, the things that got turned on were the receptors, were the signaling that says, "Hey, I am now ectoderm." So then we become pluripotent. We are ectoderm. Become determined to be neural tissue because it received the signal. And then once you differentiate, you've built something because of that signal.
Okay.
So signaling is what tells it to become something. So if we are nothing, we are anything. We have no signaling. If we're pluripotent, we are ectoderm. We are being told to be ectoderm.
Okay, that makes sense.
Is this the only signaling that's going on, or is there a lot more? Or is that a complicated question?
That's a complicated question because, yes, so we have steps, right? The steps are blastulation, gastrulation, neurulation. But at the same time, other signals are taking place. Other things are being turned on and off. Things are being histinated, non-histinated, methylated, and so forth. So it is very dynamic. It's changing constantly. So yes, lots of things are happening. But what I want you to worry about is those steps. Fertilization to blastulation, we're making cleavage. Blastulation, gastrulation, we make our 3 tissue layers. Gastrulation and neurulation, we're making our neural tissue. Then we're going to go through organogenesis. That is what I want you to worry about.
Okay.
I wanted to go through the Model 3 or Genesis just to get it better because I think I'm a little bit confused. Just the process of that's in 10, the worksheet, meiosis.
Oh, okay. So worksheet 10. And we're in Model 3, you said?
Yes, thank you.
Okay. So exactly what we did yesterday in class, in lab. And then we find all the cool shit. Okay, so I guess let's start with spermogenesis. Okay, let's go back. What's the first step between panel 1 and panel 2? Meiosis or mitosis?
Mitosis.
Mitosis, okay? So we go through mitotic division to be able to create your thing. So that's a stem cell, right? So let's go down to meiosis in oogenesis. What's our first step? Meiosis or mitosis?
Meiosis.
Meiosis. We never go through mitosis, therefore we're limiting number, all right? But do we complete meiosis?
Not until fertilization.
Not till fertilization. So what is this showing, right? The egg is getting bigger, but we're also containing all of those nutrients. So the follicle cells are growing. So between panel 1 and panel 2, we have that little circle inside. That's your egg. Then all the other little circles, and the black, those are your follicle cells. They're getting bigger. They're producing all the nutrients. They're also producing everything to push that egg out. So in the ovaries that we looked at yesterday, some of them had little yellow dots on them. That's what the follicle cells are. They push that oocyte out. So as it's growing between panels 2 and 3, those follicle cells are numbering in size. They're pushing stuff out. They're adding nutrients to the egg. In humans, our eggs stay relatively small, but in things like chickens, eggs become huge. They need a lot of nutrients. The follicle cells are helping to do that, but they're also helping to regulate hormones. Follicle cells are making the hormones that are going to tell when to do ovulation. They're making the hormones to say, hey, this is how you make a uterus and placenta and all that stuff. So the follicle cells are making a lot of stuff, and they're doing that. And between panel 6 and 7, that it's filling full of fluid and pushes it out, it erupts out. So that's what is ovulation. So panel 7 is ovulation. Now, we still have not completed meiosis 2. We still have a polar body there, and we still have replicated chromosomes, so we have sister chromatids. When we have meiosis 2 is when we have fertilization completed. And then you would see the development with that embryo, for instance, yesterday that we saw. Yeah.
Is this why folic acid is very important when you're trying to get pregnant, because it's giving helping your follicle cells?
No, actually, folic acid has nothing to do with follicle cells. Folic acid actually has to do with neural development. You need folic acid for neural development. Typically, you do not need to take vitamins and folic acid and stuff before getting pregnant. It's much more important afterwards.
Interesting.
So not even during, you say?
During pregnant, yeah.
Oh,
once
after,
you get-
duh,
yeah,
I'm sorry.
after fertilization.
I was thinking labor. I don't know.
Oh.
Sorry. Okay, so from 1 to 2 is meiosis, and then from 2 to 3 is there's growing and they're pushing nutrients. From 3 to 4, are they separating or it's forming, or could you?
Yeah, it's just getting bigger. It's just getting bigger.
Just getting
It's all
bigger.
good. 3 and 4 is getting bigger. 4 to 5 getting bigger. More follicle cells getting bigger, bigger. Meiosis 1 is completed in panel 6. Panel 7, we get the eruption of the egg. You still have your polar body sitting right there, but you still are replicated chromosomes. And then meiosis 2 completes at fertilization.
Okay.
So, why do the follicle cells grow if the oocyte is not growing? It might just be a confusion for me.
Okay, say that again. I missed something.
Because we see that the oocyte stays like the same size, but the follicles are growing every time. Why is that?
So the follicles grow in number because you just need a lot of support cells. We need a lot of support cells for the fluid, but also all those other things like I was talking about, the hormones, the regulation, everything else. Is that what we're trying to talk about?
Yeah.
That's it.
So for support, the fluid and also the hormones, right?
Correct. And so then let's talk about issues. So let's imagine the follicle cells keep growing between 6, and they keep going, but 7 never takes place. What do we call that?
That'd be a cyst.
That's an ovarian cyst.
Ouch.
So in 7, it releases, and then what's left that behind is what? Like,
So what gets left behind on the ovary forms scar tissue. That's it. It's that yellow, like pussy scar tissue. The oocyte moves into the fallopian tubes, where it'll either be fertilized or it won't.
Does there become a point where there's so much scar tissue that an egg cannot be released?
I don't know if that's true, but definitely over the lifetime of a female, the scar tissue builds.
Okay. I don't think I have any more questions. I think most of my questions are more about immunology. But
Okay,
we're gonna get to
we've
that.
done that. Yeah, we're doing that next week.
Yeah.
Cool. Anybody else?
No, I don't have anything.
Cool. Well, Taylor, you hang out for a second. We'll chat about lab.
Okay.
Anybody else?
No.
I have a side question though about applying for graduation on my CTC link. I can't apply yet, and is that just because I haven't finished this class yet, or?
No, it's not done in CTC Link. It's a Google form.
Oh, what the heck? Okay,
So
I'll look it up on the
just
website.
Google EVCC graduation.
Okay.
And it's a
Okay.
graduation process, and it is literally just a Google form.
Okay, I was trying to do it through CTC link. Okay.
And then, graduation applications in my EVCC portal. I believe it's just a Google form.
Okay. Thank you.
Yeah. They could have changed it, but I believe it's a Google form.
Okay. Thank
Anybody
you.
else apply that would know?
Yeah, it's on the thing for my EVCC, and it's on the bottom, and it's a Google form.
Yeah.
Okay. Thank you guys. Appreciate it.
They didn't change it on me.
Yeah. All right. Bye, everybody.
Bye bye. Have a good weekend, everyone.
You too.
Thank you.
Team wise.
Yo,
Thank
bye-bye.
you.
Yep.